A major contribution from our work was to change the emphasis from morphology, which is what the cells looked like in stain preparations, to function, that is, what the cells could do. And to that we added something that both of us considered very important, namely ways of finding their number, quantitation. So we changed morphology into function and quantity. At the time we started to work, there was a controversy about how radiotherapy helped patients with cancer. Many radiotherapists thought that malignant cells, cancer cells, were more sensitive to radiation than normal cells. On the other hand, there was emerging cell culture evidence that said that all mammalian cells were roughly similar in their response to radiation. And we wished to test that on an important cell, a blood cell, and we wished to test it by irradiating the cell in the animal, because the criticism had been made that all of these other studies were based on irradiating cells outside of the animal in cell culture. So that's what we were doing. And what we had done was to devise an assay for the function of blood marrow cells, and the assay depended on the capacity of marrow, normal marrow, injected into irradiated animals to protect those animals from death. So we knew the relationship between the number of normal cells that you had to inject and the degree to which irradiated animals would survive. And that was our assay. And we were using that assay to measure the radiation sensitivity of normal bone marrow. The experiment that we actually did was to inject various numbers of normal marrow cells and irradiated marrow cells into heavily irradiated recipient animals, then to kill groups of animals at times in order to examine the marrow as it was proliferating in the new hosts. One of the days where animals were killed happened to be a Sunday. I killed 10 mice and took out their spleens and immediately observed that these spleens were lumpy. Now, both Dr. Till and I had studied colonies of cells growing in cell culture. So we were very open to the notion of cells in colonies. And when I saw those lumps, I thought, is it possible that I'm looking at colonies of cells? In culture, when you have colonies, what you do is to count them. So I tried to count the lumps, and I found I could count them very easily, and that there was a relationship between the number of normal marrow cells injected into the animals and the number of clumps. And moreover, that relationship was what we call linear. That is, put in 1,000 cells, you got one lump. Put in 2,000, you got two lumps. And you could plot that. And before we could go any further, we had to answer a crucial question, which is whether or not these colonies that we were counting as lumps were in fact clones, that is, derived from a single cell. And to answer that question, Andy Becker, working with Jim Till, used a chromosome technique to demonstrate that in fact each one of those lumps was a clone derived from a single cell. And therefore it was possible to use the lump as a way of deducing the characteristics of that single cell. And that observation of Andy Becker's was key to what we were going to do. There was a good deal of interest that I don't think people were surprised. They were rather pleased that this new assay had become available, and it brought together two groups of people. There was a group of people centered at Oak Ridge National Laboratories who were interested in marrow as a radioprotective tool. They were interested in radiation protection. You have to remember this was the time when everybody was afraid of the possibility of nuclear war. And then there was a group of hematologists who were interested in blood as the site of important diseases that they were studying. And those two groups in general worked apart, but the technique, the spleen colony technique that Till and I found, was of equal interest to both and served to bring them together. I was very fortunate to hear Dr. Tibby Russell give a paper on genetically anemic WWV mice at a meeting, and screwed up my courage, and it took a little to go up and approach her afterwards for conversation, and she was the soul of pleasantness, and we talked for hours, as a result of which she sent us some WWV mice, and we tested their marrow and found that it did not form colonies. So we said that the W gene caused a defect in stem cells that prevented colony formation. And later we had, again in collaboration with Dr. Russell and her collaborator, Dr. Selden Bernstein, we examined a different strain of mouse that appeared superficially to be very similar, the SLSLD mouse, which is also white with black eyes. And when we tested marrow from that mouse, we found it formed colonies perfectly normally. But if you irradiated SLSLD mice and injected them with normal marrow, it didn't grow. So we now had a pair. We had the W mouse with its defect in stem cells and the steel mouse with its defect in the environment, which allowed the injected cells to grow. That work is extended now so that there's much interest in the actual W and steel genes. That they have been identified. They have been studied at the molecular level and are a subject of great interest and importance to this day.